Thanks again.
Thank you very much for the invitation to speak to the Center for Computational Biomedicine at Harvard Medical School. And before we start today's talk, I would like to tell you a little bit of my background and how I fell in love with semantic similarity. Because as Rafael said, this has been a more than a decade-long affair. And so I'm a biologist by training. My undergraduate studies were in cell biology and biotechnology. But when I went into the lab for my internship, which was a really interesting internship in genetic engineering of rice, I actually found myself less and less attracted to the immense hours of pipetting that I needed to do back in the day, and more into the computers and data analysis side of things. And so I decided that I would turn my life around, and my desire of becoming this biomedical and molecular biology researcher would be transformed into becoming a bioinformatics researcher, which at the time was a fairly new thing, especially in Portugal. And despite many people telling me bioinformatics is going nowhere, it's not going to be a research area, you will not be able to do a PhD on it, I persevered and I did. And I remember very vividly, and this was back in 2006, when I first saw my first presentation about ontologies and the gene ontology. And I thought, oh my God, this is the perfect thing for me, because this marries all of my very diverse or eclectic interests, because I loved biology, but I loved computer science. I also loved philosophy and linguistics. And so I thought, here is something that touches all of these very interesting and diverse areas that I have been told that they have nothing to do with each other, when in fact they do. And so that was the beginning of this work. Of course, today I'm going to tell you about more recent work, so papers that were published in the last couple of years, but all of this goes back to that day in that classroom where I first learned about the gene ontology, and I found my passion for this area of research. So today, the title of the talk is Semantic Similarity for Humans and Machines. Although for the better part of the last decade, I focused more on semantic similarity for machines and not so much humans. It was semantic similarity designed by humans to be used by machines. But in the last couple of years, I've been doing this reverse, designing the semantic similarity using machines and then having them be more useful and helpful for humans in a more understandable way. And so this is going to be a story from there and back again for humans and machines with semantic similarity. So I'm sure that I don't need to tell you that measuring entity similarity is a key aspect in many applications in the biomedical domain, and especially in bioinformatics. And we have all of these techniques for comparing genes and cells and organisms and populations. And some of them are more easy to do. So for instance, we can apply sequence similarity algorithms that just by looking at the sequences of nucleotides in the sequence, you can get a score that reflects the sequence similarity. But when we want to compare more complex biomedical entities, for instance, a disease, it is not clear exactly which aspects should be taken into account, and more specifically, how we are going to describe these aspects in a way that is computationally amenable. So how do you describe a headache or muscle pain or high fever or sneezing? Of course, maybe that you could tell me, okay, we can use language, right? We use language to communicate with each other, so we could just compare the words that describe these concepts and use them to find some kind of similarity. But words are tricky. I'm sure if you have worked in the biomedical domain for quite some time, you know that we are plagued by two pests, synonyms and homonyms, okay? So two different names for the same thing, or the same thing being called by two different names. And although more recent language models that you see in the news, the deep learning language models, are getting better and better at this, they still struggle with understanding more complex phenomena that are quite rare in the data sets that they are trained on. So we have this challenge of how to measure the similarity of these complex entities. And one of the ways to do that is, of course, with my passion, using ontologies and knowledge graphs, which are structures that combine the ontologies with a data they describe. And we can use these structures to compute what is called a semantic similarity. So a similarity in the meaning of the entities. So I'm just going to give you a brief overview of a very simple overview of what an ontology is. But if you already know all that I'm going to say, please stop me in my tracks and I can move on to the more specific parts. But just very quickly, so an ontology describes the concepts that exist in a domain. So for instance, each of these nodes in the graph is going to be a type of biological process that is used to describe a protein. Each of these ones is going to be a cell component where the proteins are active. And these will be different types of molecular functions that proteins perform. So this is actually a very simplified representation of the gene ontology, because actually gene ontology is over 40,000 classes. So over 40,000 of these nodes that describe different properties of protein function. And ontologies have a very strong taxonomic backbone. So there is a hierarchical structure between concepts that are more specific and concepts that are more generic. So by having this hierarchical structure of the concepts and being able to link the proteins to what concepts they are described with, we can use them to compute the semantic similarity. And semantic similarity can be computed for entities of the same type, but we can also extend this to compare entities of a different type. So for instance, we can compare the phenotype of a disease with the phenotype that a gene elicits. So in this case, we have here a very summarized version of the human phenotype ontology that describes phenotypes. And we can use the ontology to describe both diseases and genes through their phenotypes. And then we can compare different entities as long as they are described in the same semantic space. Most commonly used semantic similarity measures are what we call taxonomic semantic similarity measures. This means that they make use of this hierarchical backbone of the subclass and class relationships that link the more specific concepts to the more generic ones. Very simple ways to measure the similarity between the two entities that are described in the ontology can be made by just looking at the shared portion of the graph that is induced by the descriptions of the two entities. So in this case, we have protein P1 and protein P2. And you see here in this darker purple, what is the shared portion of the graph that they have. And this shared portion can be used for a simple ratio in this example that gives you a measure of how similar they are. Of course, this is a very basic measure of similarity. Expertly designed algorithms can grow a lot more complex than this, but an important aspect of all of these algorithms is that they usually take into account how specific a concept is, and more specific concepts have a larger weight in the similarity. So if you share very generic description, you will not have a high similarity, but if you share a very specific definition, then you will have a higher similarity. And so for more than a decade, a very large number of semantic similarity measures was created that explore these taxonomic relations in the ontology and this concept of specificity of the concepts or classes in the ontology. And they were used in a number of applications, from predicting protein-protein interactions, validating prognosis, clustering for protein function prediction, a number of different applications. But one of the things that first inspired us to change a little bit of this paradigm was this reflection that we have actually these very large biomedical ontologies, such as the gene ontology or the human phenotype ontology, tens of thousands of classes, and they usually don't just stick to one perspective when describing a biomedical entity. So for instance, in the gene ontology, you have these three aspects that are rooted in three different roots in this ontology graph, which are the biological process, the cellular component, and the molecular function. So these three different semantic aspects give you different perspectives over the knowledge graph entities, that is, the entities that are described under the ontology, and they allow you to consider different types of similarity potentially. So we have on one hand this idea of a semantic aspect being this different perspective of a description of an entity within the ontology using a subgraph, but you can also extract different semantic aspects by building the graph just looking at specific types of relations in the graph. So for instance, we could have a gene ontology graph that only uses the regulates, positively regulates and negatively regulates relations. And so that would give you a different perspective on the similarity rather than just looking, for instance, at the ESA relations, which are the taxonomic backbone that is usually used by most semantic similarity measures. So this idea that we have these different semantic aspects was one of the things that first inspired us. And the other was that so far semantic similarity was mostly used with two purposes. One, to measure a similarity and to show to a user how similar two proteins are, or two patients with a given phenotype are. And so they were very much user-oriented. You are showing the similarity to a human end user to support some kind of decision, either to choose a candidate gene or to recommend some kind of therapy to patients, and also using it to validate other bioinformatics approaches, to validate clustering, to validate supervised learning. So it was not used as a feature per se for data mining aspects. It was mostly for users or for validation purposes. But our idea was that actually we can have semantic similarity for machines. And what I want to say with this is that semantic similarity can be a feature used in machine learning. Okay, so that's the play on the words. So when we go back to ontologies, we have this challenge in how we are going to represent the information in an ontology or in a knowledge graph in a way that machine learning methods can consume. And most machine learning methods consume vector-based data, and an ontology is a graph-based data. So there is this need to learn representations based on the ontology that then can be fed to the majority of these machine learning algorithms. And we have two state-of-the-art methods to do this. So an older version is graph kernels that learns patterns in the graph. A more recent version is representation learning through graph embeddings, where you learn a vector that approximates the properties of the graph. But our hypothesis was that in some specific cases, semantic similarity itself could be the representation, could be the feature generation method that we would use for machine learning applications. And so that brings us to the first paper that we are going to discuss today, which is Evolving Knowledge Graph Similarity for Supervised Learning in Complex Biomedical Domains. And what is the question that we start with? So the question is, can we evolve a semantic similarity measure that is going to fit a particular learning task? So we want to use semantic similarity directly for a learning task, but combining it with a machine learning method that is able to evolve it, to adapt it, to tailor it to a specific learning task. So up until then, the traditional way of doing things is that an algorithmic semantic similarity score would be computed. Perhaps it would be fine-tuned for an expert to support a learning task, and this would rely on the user's intuition or experience to do. And our idea here was, okay, that maybe we can, one, go back to the idea that we can have those multiple semantic aspects, those different perspectives in the graph, use those as ingredients to a genetic programming model that can learn to select the best combination of these different semantic aspects that is going to fit a specific learning task. And so our approach is we take a graph, we define what are the semantic aspects that we are going to consider. And in this work, we consider the three VOSA graphs, so the biological process, molecular function, and cellular component. We computed a matrix of all similarities. And we use this for protein-protein interaction prediction. And so we retrieved positive examples from interaction databases, generated negative examples from random negative sampling. And then we evolved this genetic programming model that was able to tailor a combination of these different aspects of similarities and then use it to predict protein-protein interaction prediction. And how does the genetic programming part work? So genetic programming is a machine learning method that is inspired in Darwinian natural selection. We have an initial population, and this initial population is a population that is made up of the features that we have to build an answer and operators between those features. So for instance, in this work, we used the maximum, the multiplication, sum. So we used a number of mathematical operations and other features. And then the best combinations of these features pass through selection. Those that are the best ones go forward to the genetic operators of crossover and mutation. So different bits of the solution are combined and mutated, going back to selection until after a number of loops here, depending on our stop criteria, we have a winner solution. And what does a winner solution look like? It looks like a tree where the different features are combined using operators. So for instance, this means that the maximum between the feature 0 and 1 is multiplied by the maximum between feature 0 and 2. And when we did this with our 3 semantic similarity scores, one for each branch, we applied it to a data set. We built a knowledge graph from the gene ontology using the gene ontology and all of the annotations that describe proteins in the gene ontology annotation database. And we use a number of different benchmarks for protein-protein interaction data sets. These covered a number of different species and came from a number of different databases. And we have a large variety of data sets, both in terms of size, in terms of richness of annotations, and in terms of origin of the species. So we have very large data sets with 31,000 plus interactions and very small ones with just 550 interactions total. And so then to compare, what we wanted to do was take what was the standard modus operandi, so the traditional way of doing things where you would just compute the similarities and combine them with either the average or the maximum, or just take one of those similarities. And we simulated this expertly tuned semantic similarity by selecting the best possible threshold. So we gave our baseline this huge advantage that we selected the best possible threshold to predict protein-protein interactions just based on the similarity. And then we compared with our evolved semantic similarities. And so these are the results for this comparison. And what we see is that even when we combine the different aspects using either the average or the maximum, in most cases, in 7 out of 9 data sets, our evolved combinations that use genetic programming to learn that formula, how to combine the different aspects, outperforms this just using a single aspect or a combination of single aspects with expertly, optimally fine-tuned thresholds. So in these 2 data sets, we don't get as good as a combination of single aspects, but in most of them, we do. But then we asked a different question. What if we did this prediction using a pool of the data sets that belong to the same species? And what we saw and what was more interesting is that the combined data set for human actually perform better than either two of the separate ones. You might be expecting that the combined performance would be something in the middle, but actually the information is complementary between the two data sets, and we can actually train better models to evolve a semantic similarity for the human data. So for instance, for the yeast combined data set, you don't see that. So the yeast data sets are these three, and the combined one has slightly lower performance, but for human, we see the exact opposite. But all in all, you see that for most data sets, you have really good performance, so above the 70s in most cases. Human is a little bit harder, but if you combine the data set, you can have an F measure above 75 for human. The other question was, okay, what if we trained on all of the other species and then just tested on one of them? So if we're testing on fly, it means we trained in E. coli, human, and yeast. And what we found out is that when we have a model that is trained on multiple other species, we can, with some success, generalize this model and apply it to a different species. So it is possible to produce this species agnostic model that learns an evolved semantic similarity tailored for protein-protein interaction prediction. Now let's compare with training specifically for the same species we're testing or these generalized models. So for fly, we lose just a little bit of performance, a little bit more for E. coli, somewhat more for human, but for yeast, you really don't lose a lot of performance by using these generalized models. And so we can have this model that successfully predicts for all species. And when we pull everything together, we have an aggregated F measure of 78. So one of the things that we were interested in is in seeing the frequency of the variables, the three semantic aspects, biological process, cellular component, and molecular function in our learned genetic programming models. And what we saw is that the most frequent variable to be present in the models, whether it's in the single data set or in the species agnostic trained model, is biological process, then cellular component, and finally molecular function. And this should not be surprising, okay? There is a reason we are studying this in protein-protein interaction, is because we know that there's this expectation that to interact, two proteins should participate in the same biological process. They should be present in the same or adjacent cellular components, but they don't necessarily need to perform the same molecular function. So the frequency of these variables reflects what would be expected. So at the end of that work, we had these three ideas for future work. We wanted to add more semantic similarity measures to the evaluation. We wanted to see if we could apply this to different supervised learning tasks. And we wanted to test other semantic representations, not necessarily taxonomic semantic similarity, combined with this idea for semantic aspect selection. And the first thing that we decided to tackle was the application to different supervised learning tasks. And that brought us to a challenge, which is how can you evaluate semantic similarity in different tasks beyond just the protein-protein interaction prediction? And so that brings us to the second paper, a collection of benchmark data sets for knowledge graph-based similarity in the biomedical domain. So before we move on to this paper, I would like to ask you if you have any quick questions regarding the previous paper.
I have a quick question. So you said your similarity also involve target to the learning task. So is it also a learning process you're saying there?
Yes. So we use the genetic programming, which is a machine learning algorithm, to learn those formulas, those combinations of the operators, the maximum, the multiplication, the sum between the different features. And so let me just go very quickly back here. So this is a result of something that was learned, okay? So the model is this. It's a very simple combination of different operators with the variables. Okay, so this is one of our simpler models. Models were actually much larger than this, but this is an example of a result from tailoring, from learning how to combine the different features to produce this tailored semantic similarity measure that is able to perform well in protein-protein interaction prediction.
Okay, thank you.
Any more questions? Okay, I'm sure you will have more by the end of this. So the challenge of evaluation. At this point, we have over a decade of works in semantic similarity algorithms, but there are very few studies for the evaluation of these measures, and mostly they are very small scaled. So we had these three goals that we wanted to tackle with this work. We wanted to be able to build a benchmark that not only includes different species, but also different knowledge graphs. So far we have been working just with proteins and a gene ontology, and we wanted to expand this. We wanted to support a large scale evaluation of semantic similarity measures. So we wanted our data sets to be fairly large. And we wanted to support proxy-based evaluation of semantic similarity measures. So bear with me, I will explain what this means in a bit. Bear with me and I'll get you there. So one of the problems in evaluating semantic similarity is that depending on the use case and depending on the user, the assessment of similarity can be quite different. So in this example, these two proteins, adenosine kinase and pyruvate kinase, they can be considered similar or not similar depending on the application. So for instance, for a molecular biologist who is looking at secondary structure, they can be considered not similar. For a biochemist looking at molecular function, they are both kinases, so they are similar. And for a physician, since their malfunctioning causes different diseases, they are not similar. So domain experts can be biased in their assessment of similarity, and it is not always straightforward to understand the reasons behind a given assessment of similarity. By this point, there were a few very small benchmark data sets where experts had indicated the similarity for different biomedical entities, but there was no justification behind that decision for similarity. And as we see in this very small example, the justifications can give rise to very different appreciations of similarity. So our idea was to, instead of looking at the domain expert opinion, to look at proxies of similarity. And so we decided to target three proxies of similarity. So one is what you would expect by now, because it was one of our examples at the start of our conversation, which is sequence similarity. The other, of course, is protein-protein interactions that we had used in this previous work. And then we also looked at molecular function similarity given by the number of shared PFAM domains. So by looking at these three different types of similarity proxies, we had an idea of measuring the similarity between proteins from three different aspects. And so our methodology to build this benchmark first was to ensure that our entities that would be part of this benchmark would be sufficiently well annotated to improve the reliability of the semantic similarity measures. So we decided to include proteins that would have at least one annotation to one of the very specific Go classes, and other proteins that would have at least one annotation to a very specific Go class in all of the three branches, so the biological process, molecular function, and cellular component. Then we built pairs between these well-annotated proteins, and we decided to select these pairs looking at their gamut, their spectra of similarity values according to the proxy. We wanted to have a fairly good distribution of molecular function similarity, presence or absence of interactions, and sequence similarity. And so we use these to build our final data sets. We also extended our benchmark to also include the human phenotype ontology, and the gene roles in phenotypic abnormalities. So for this, we used homing phenotypes for these genes in the human phenotype ontologies as our proxies. So we built data sets for protein-protein interaction prediction, where we had the proxy of protein interactions, and we also included sequence similarity because we could. Then protein molecular function with the similarity between the domains and also the sequence similarity. And then finally, the gene role in phenotypic abnormalities. We included a number of different semantic similarity measures. And here we wanted to combine what were at the time the most representative measures of the different approaches of the state of the art. So we chose two different ways of measuring the specificity of the classes. So if you remember, that is a very important component in measuring taxonomic semantic similarity. And we have IC-SECO, which is a structural measure of specificity, and IC-RESNIC, which is called an extrinsic approach because it looks at the number of entities that are annotated with a class to measure its specificity. If an ontology class describes a very large number of entities, then it is considered a very generic class and doesn't really tell you much about the similarity between those two entities. And then we also combine this with two different approaches for entity similarity. So the SIM-GIC measure, which was actually, this was my first work in semantic similarity all those years ago. So this is simply a ratio of the shared portions of the graph that is weighted by the information content. So more specific classes have a higher contribution for the similarity as calculated by this Jacquard similarity ratio. And then finally, we also use the best match average, which just computes all of the pairwise combinations of the similarities between the different classes that describe the two entities, and then just makes the best pairs, and then just sums the similarity for each pair in each direction, making the average. So the similarity from A to B and the similarity from B to A. So we have these representative measures. We have all of those proxy similarities based on the gene ontology and the human phenotype ontology, and we are missing the data. And so the data, we have data sets for E. coli, Drosophila, Homo sapiens, yeast, and all of the species combined. We have data sets ranging in several numbers from, a few hundreds to over 158,000 pairs in our data set. And then we also have the data set for the human phenotype ontology, which is just for Homo sapiens, with 12,000 pairs. So we have this larger scale benchmark of data. So now you have a way to evaluate the semantic similarity measures. So one of our future work ideas was tackled, but the other one was to include different types of semantic representations into this training. And as we discussed in the beginning, a more recent and popular type of semantic representation based on the graphs of ontologies or knowledge graphs or knowledge graph embeddings. So in very straightforward and simple terms, a knowledge graph embedding is a low dimensional representation. This usually means it's just a vector representation of a few hundred entries in the vector. And machine learning methods or matrix factorization methods are used over the graph to produce these vector representations that retain properties of the graph. So if you compare these two vectors that are representing in this shared embedding space, two similar vectors here will correspond to two very close nodes in the graph. And so then you can represent each entity in the graph by a vector. And by comparing the two vectors, for instance, with something as simple as cosine similarity, you can get a different type of semantic similarity that is based on the knowledge graph embeddings. So we wanted to extend our first work with these different kinds of representations and more semantic similarity measures. And so we developed this framework, EvoKGSim Plus, that tailors knowledge graph-based similarity for supervised learning. It's available on GitHub, and on this webpage, you have an interactive description of what the framework does and the results that we achieved. And just to give you a sneak peek, we evolved both the classical, the taxonomical based semantic similarity measures and the knowledge graph embedding similarity measures. And we used a number of different algorithms for knowledge graph embeddings. And what we found out is that actually still the classical measures perform better in protein-protein interaction prediction, although some of the knowledge graph embeddings methods also perform quite well. We were not expecting this because we were actually expecting that the knowledge graph embeddings would be better because they look at all kinds of different relations in the graph. Some of them are based on random walks in the graph that have a length of 8 hops in the graph. So we were expecting them to be much better. So they afford actually a representation that should have more detail. But in fact, the semantic similarity, the classical ones, tailored algorithms, were still working better for this application. So this brings us to the final part of the talk, which is the other way around. So we have been talking about semantic similarity for machines, how we can use semantic similarity to generate features for supervised learning. But there is also this unique opportunity of ontologies and knowledge graphs for explainability, which is obviously very hot topic right now in AI. And the basis for this idea is that we have the semantics for the data entities in the ontologies. We know what each class means. That meaning is given by the ontologies. But then all of the machine learning methods that are very hot nowadays, working over graphs, they just use knowledge graph embeddings. Knowledge graph embeddings are not explainable. So when you build those vectors in that low dimensional space, each feature of the vector has no meaning, okay? It is just a compression of the semantic space into those dimensions. But semantic similarity is explainable. When you measure the semantic similarity for a specific aspect, you can understand that that means that those two proteins are in the same or a very similar cellular component in the cell or perform very similar molecular functions. So there is this unique opportunity for combining the explainable features that semantic similarity gives you with interpretable machine learning models. And so genetic programming is potentially interpretable, but it is not always humanly interpretable. So this is the example of one of the models that we obtained in our first work. And I mean, you could read this, you could, but could you really understand it? I don't think so. So these models may grow exponentially with each generation, and this potential for interpretability is lost. And moreover, there is this added challenge that some of the operators, for instance, such as multiplication and division, are not very clear what they mean in biological terms. What does it mean to divide your biological process similarity by your cellular component similarity? So we came up with this modified version of genetic programming that would first penalize solutions that would grow very large, and then we would restrict the type of operators to operators whose meaning is a little bit more clear, such as the maximum, the minimum, the addition, and subtraction. And so this took care of the interpretable model part. So now we have models that are small enough to be readable and with operators that can be more easily understood. But the next part is we have been talking about working with 3 features: biological process, cellular component, and molecular function similarities. Those are not very explainable. Those are very generic. And so our hypothesis was, if we increase strategically the number of semantic aspects that we use for these models? Can we improve the explainability of the models? And how does it affect the performance of the models? And so we actually tested not just our 3 original semantic aspects, but also 43 and 52. So 43 are the direct subclasses of the Go root, so the classes at this level, or the whole graph, but that are not leaves, so they have children. And 52 is just the direct subclasses, including those that where the graph ends there. They are leaves of the graph. And so by combining these two aspects, using more semantic aspects and using more interpretable machine learning models, we were hoping to be able to produce models that can be understood and that don't sacrifice too much the performance compared to our original work. And so what we found out is that although there isn't a lot of gain in performance by increasing the number of semantic aspects, there is also not too much of a loss. And more interestingly, by restricting the size of the genetic programming models and using just the more explainable operators, we could still get fairly high performance compared to our original models. But the median number of nodes in these larger solutions was much smaller. So now we have a median number of 17 nodes compared to 49 in our aspects. And I'm going to show you one of these models. So this is a model that predicts protein-protein interaction, and it is actually quite easy to read. So it is a maximum of the similarities based on these aspects, so catalytic activity, cellular process, etc., the signaling, plus an addition of these two, behavior and immune system process. So we now have models that can be more easily understood and that don't sacrifice too much in performance. Of course, one may argue that we don't need this for protein-protein interaction prediction. I've heard this before from other people, and my comment is twofold. First, why don't we need it? And second, and perhaps more interestingly, which is, if you want to make an explainable method, it's good if you start testing it in something that can be explained actually, and where you can evaluate if the explanations make sense. So the understanding of this explanation is that two proteins that interact usually participate in the same types of biological processes. But now I'm going to show you something more interesting than just looking at the F-measure scores. So these are two examples of two correct classifications of our GPCX43Go model. And so what you can see here is that these two proteins and these two proteins, you have here the semantic similarities for a number of different aspects. And what you can see is that they have fairly high and very high similarity for a cellular and anatomical entity where they are. And so they are exactly either in the same place or in very similar places. And then in this case, you also have a high number of different semantic aspects related to processes that also have a very high similarity. And in here, you have a cellular process that is very short, very low, and no other similarities. So here you have exactly in the same place, doing very similar processes, and here you have in a similar place, but doing something completely different. So this is a true negative, and this one is a true positive. But more interesting is to see when it fails. So here we have a false negative. And our false negative, the prediction is, okay, it's in the same place, so it's probably working together. But actually what we found out is that these two proteins don't have a sufficiently rich Go annotation, and they actually are interacting, but there are no biological process annotations that reflect these shared processes that the proteins have in reality. And finally, we also found an interesting case And so these were we just took 40 random cases and analyzed them, and these are just the interesting bits. And so this is a false negative, actually, sorry, this is a typo. So in our random negative sampling, these two proteins are identified as not interacting, but we predict that they do. Why do we predict? Same localization, a number of different processes that are very similar. And what we found out is that the literature actually describes interactions between very similar proteins that belong to each of the same families of these two target proteins. So probably this is not a true negative, it is just an unknown at the time that the database was built. So to finish, semantic similarity in our idea supports explainability. So that is the part that is important for humans, and adapts to specific supervised learning tasks, which is the part that is important for machines. Before I finish, I would like to acknowledge my PhD student, Rita Souza, who was a co-author of all of these works, actually the main author of all of these works, and she's here in the audience, so she can also answer some questions that I don't know the answers to. And her co-supervisor, Sara Silva, and also Carlota Cardoso, Sebastian Puller, and Daniel Faria. And just to finish, so this is my small research group, the Lisbon Semantic Data Lab. And contact us if you want to learn about open positions and collaboration opportunities. Thank you.
Thank you very much, Katja. Round of applause. I know it's inaudible these days via Zoom, but it's still there.
Thank you very much.
So I'll open the floor for questions, but I have one to start with. Meanwhile, feel free to raise your hands. I just wanted to ask about the semantic aspect. So a couple of questions around there. Are these parametrizable or are they learned? And then the second question is, do these represent sub-hierarchies of the ontology, or do they delve into non-is-a type relationships, so object properties in the ontology and whatnot?
Okay, so the original definition that we proposed for a semantic aspect can be either a subgraph or an overlay of the graph that only looks at specific properties. But in our work so far, we have only looked at subgraphs. In the works that we have published so far, we parameterize the semantic aspect. So we define rules to create those semantic aspects and then extract the subgraphs rooted on each aspect to compute the semantic similarity. But Rita is actually now working on learning those as part of the task. So having it be more end-to-end instead of having those parameterized to begin with.
Right, great, that makes sense. Thank you.
Thank you.
Let's see, any more questions from the audience?
Yeah, Raphael, I have one. Great talk, Katja.
Thank you.
Just wondering how you think about, sort of the circularity problem, right? A number of the data sets that you're using to assess similarity in the gene ontology were actually used to create the gene, right? I mean, it's a lot of protein interaction data went into assessing especially biological process. And I think sequence went into assessing similarity of molecular function when they were doing it. So just curious how you guys think about that.
So two things. So regarding the sequence similarity, so that is the easiest one perhaps to say, is that we look at different evidence codes for the annotations and remove some of those that would be responsible for circularity. Of course, we can't be sure that that doesn't exist in some way, but that's how we handle it. But we actually were very interested in that question regarding the protein-protein interaction data sets. We were very worried about data leakage. And so I'm not presenting it here, but Rita did a very interesting work that actually won Best Poster last year at the conference, where we were investigating if there was data leakage between the gene ontology and the protein-protein interaction data sets, and we did that by training our models in older versions of the database of the string database and testing in new versions of the string database and see how that affected the performance. And actually what we found out is that the performance is comparable, whether you are training and testing with the same version or training in a much older version and testing in a new one. So there may be still data leakage, but it doesn't seem to be influencing the performance of the methods. How that influence explainability, it's still an open question for us.
Great, thank you.
Thank you for the very interesting question.
Great. If I don't see any more hands up, I'll keep asking questions. I have another question, which is about just getting your thoughts on knowledge graph embeddings versus semantic similarity. So we've seen a lot of activity around knowledge graph embeddings these days, and I take your point, indeed, they're not explainable. Just wanted to have an idea of how, in terms of computational complexity and feasibility, how practical would it be for one to adopt a semantic similarity-based approach versus a knowledge graph embedding-based approach? Did we have enough support there? What are sort of are there any sort of sticking points that should be solved before that becomes practical, or what do you think about that?
Okay, so I think that there are two sub-questions, so I'm going to try to talk about those two topics. So one is this idea of computing the knowledge graph embeddings and then using the vectors to compute the similarity, and then you just have a similarity which is equivalent to using a classical or more traditional algorithmic semantic similarity. It is more costly to do that because you need to learn the embeddings, and running the algorithms, it's usually computationally much less expensive. However, if you already have embeddings learned, it's just a cosine similarity. It's quite simple to do. The other aspect is comparing using the knowledge graph embeddings as directly your features for instance, predicting the protein-protein interaction versus using the semantic similarity as your feature. And we don't have those results published, but surprisingly, semantic similarity still works best, especially when we have the multiple semantic aspects. So I'm not going to say that this is something that solves all problems. We've tested this in protein-protein interaction prediction and are testing it in gene disease association prediction. And so far, our discoveries are that. And I think that actually one of the possible explanations behind this is that the knowledge graph embeddings methods are designed for knowledge graphs, which have small schema, very large number of instances. And we are working with something that, okay, we do have a lot of instances, but they don't have relations between each other. They just have the links to the schema, to the ontology part. And so a lot of those computations for the embeddings are working on an ontology and not on a knowledge graph. So it can be possible that there is this mismatch between the way that knowledge graph embeddings that were designed for very large knowledge graphs that have a shallow semantic component versus using them on a full-fledged ontology. So that is also something that we are interested in pursuing.
I see. Great. Thank you so much, Katja. Question from Benjamin. Go ahead.
Hi, it's nice talk. I'm Ben Yuri, and I was wondering, like you mentioned a lot about using these semantic similarities for protein-protein interaction prediction. And can you talk a bit more about some other tasks that these might enable? Like what other applications there might be that you've thought about?
Okay, so I can tell you about some of the work that we have been using semantic similarity for. So we have been using it to predict gene disease associations. We have been using it to compare patients in terms of phenotypes, clinical procedures, etc., to find similar patients. So for patient stratification, we have used it in the validation of text mining results for named entity recognition tasks. And that is just in my lab. And I've also used it to compare patients described under a number of different controlled vocabularies in electronic health records to predict antimicrobial resistance susceptibility. So we have all of these different applications for it, but beyond those applications there are a number of applications for comparing entities. So for instance, protein function prediction is one of them, although we don't work on that. Candidate gene prediction, patient stratification, diagnosis of patient based on phenotype similarity. There are a number of different applications that are out there.
Thank you.
Great, thank you. Let's see if we're at the hour. So unless there are any other questions, any other raised hands, I think we're going to call it a day. So going once, going twice. Great, thank you so much for giving us this wonderful talk, Katja. It was great to have you. If there are more questions, I think we can follow up via email. Otherwise, I would like to say thank you again. Thanks everyone for joining and hope everyone has a great day. Thank you again, Katja.
Thank you for having me. It was my pleasure. Thank you very much.
Thank you, Katja. Thank you so much.
Thank you, Katja.